Hey everybody, I'm really sorry about that. That was something that shouldn't have really happened then. Trying to get a meeting in the afternoon after lab finished, but for some reason, the head honcho decided do it now. Anyway, that went on way longer than it should have, and we're kind of way behind time as a result. So I'm just going to chat for like the next whatever that would be, 17 minutes or so, and we'll just need to catch up on time on Thursday because I don't want to kind of go over and then push lab later because I think that people just be too wiped out for lab. Lab is going to be a tricky lab today too. So let's get back to things. Yeah, there we go. All right, so we've got the lac operon, right? We have our structural genes, the things that do stuff, which are, where's the picture of the lac operon? Here it is. Right, so we have our three lac genes, lac Z, lac Y, and lac A, right, which make these products and generate glucose. We also have our lac I regulator that's going to be over here. And this will bind to the operator in the absence of lactose. Let me bring that up in chat. Straighten my back so I don't get a backache. So basically the idea here is you don't have lactose around. Well, you don't want expression of the lac operon, right? I mean, that is kind of a given. It would be a complete and total waste of energy making all the structural genes from the lac operon when there's nothing for them to actually work on, right? So in the absence of lactose, no lac operon expression at all. And lac I, the repressor protein, does that, and that's kind of shown here. Lac I does that by binding to the operator and preventing RNA polymerase from getting in there. So if RNA polymerase can't access the promoter, no transcription. So this is how bacteria shut off all expression from the lac operon in the absence of lactose. Doesn't matter whether there's glucose present or not. Even if there's no glucose around, it doesn't matter because there's no lactose. So there's no point expressing the structural components of the lac operon. Particularly if you don't have much energy around, you want to save all the energy that you can. So unsurprisingly, guess what the inducer is for the lac operon? It's going to be lactose, right? So if there's some lactose present, now the inducer, lactose itself, binds to the repressor protein, to lac A, lac I, sorry. And so this is the inducer, hence an inducible transcription setup. Right, so if LAC-I can't bind to LAC-O, the operator, then RNA polymerase is now free to bind the promoter and transcribe the operon. So this is the big on-off switch. No lactose present, pretty much no transcription. There are a teeny tiny little bit maybe, but effectively no transcription. Lactose present, up to 1,000-fold more transcription. It's a huge difference. You know, between 100 and 1,000-fold more transcription in the presence of lactose. Because that gene, being a bacterial gene, will be expressed in the absence of anything binding to the operator. The default state is on. So that's the big switch. What about the fine tuning? Right? Remember I told you that there's always two parts to the control of gene expression in bacteria or transcription. You've got the big change, and then you've got the little kind of fine tuning change. So what product do you think you would use as part of the fine tuning? Right, so we want to import lactose into the cell and turn it into galactose and glucose using the products of the lac operon, because we want glucose. So what part of this whole shebang do you think we would want to use to fine tune expression of the lac operon? What would tell us that we're getting enough of what we need, we're doing a good job of converting lactose? What thing would we care about if we were a bacterium?
The cell membrane?
No, that's kind of what the lactose goes through. Good idea. What's the product of all of the stuff in the lac operon? What gets produced? So if the aim is to get
Galactose?
going again,
Is it the galactose because in the lac operon there's no glucose?
So almost. But what's
Almost.
the thing that bacteria really care about? It's not galactose out of those two. It's going to be the glucose, right? So when glucose levels start rising, that's telling the bacterium that, hang on, you know, we're doing our job of converting lactose. Maybe we don't need quite as much lac-operon expression. So glucose is part of the fine sensing deal, the fine tuning of the lac-operon. Now, the way that this is done is actually a little bit complicated, as one would expect, right? So basically, we can't detect glucose directly. We have to use kind of like a sensor, essentially. And for energy levels, i.e. glucose, that sensor is called cyclic AMP. Or adenosine monophosphate. It's kind of a funky molecule. It's really just used for that. And cyclic AMP levels are inversely proportional to glucose levels. So lots of glucose equals little cyclic AMP. No glucose equals, oops, if I can spell that right, equals lots of cyclic AMP. So what the bacterium does is it detects the levels of cyclic AMP. And it does so through this thing called the catabolite activator protein. Oops, I don't want no screenshot. Go away. And so that catabolite activator protein, or CAP, is the sensor. It's what detects cyclic AMP. And it does so through essentially modulating how well RNA polymerase binds to and transcribes the lac-operon. Hey, love, Lucinda's here for you. All right. So high cyclic AMP equals active CAP equals more transcription. Actually, let's put in glucose in there. I put love glucose. That is a Freudian slip. See you soon, love. High glucose equals no CAMP, low or low. That equals inactive cap equals low to little transcription. So basically, the more glucose present, the less lac-operon transcription you need. And cyclic AMP is the means by which you figure that out as a bacterium. So to look at that in a kind of pictorial sense, and this will bring us to the end of the lecture for today. And remember, lactose is present. So lac-I has been induced to leave by the presence of lactose. So the lac-operon is essentially fair game, right? It can be transcribed. So in the absence of anything else, RNA polymerase will transcribe the lac-operon, just not very effectively. So you'll get transcription, but at a low rate. To get full transcription, you want lactose to be present and glucose to be absent, right? You need those two things together. Otherwise, you're still wasting your energy as a bacterium. So when you have lactose, lac-I doesn't bind. You can have transcription. When you don't have glucose, you'll have lots of cyclic AMP. And cyclic AMP, when it's bound to the catabolite activator protein, equals transcription. Lots of it. And this isn't an on-off deal either, right? If you have intermediate levels of glucose, you'll have intermediate levels of cyclic AMP, and sometimes CAP will help RNA polymerase, sometimes it won't. So you'll have an intermediate level of transcription. So this is like homeostasis in a sense, right? You're kind of maintaining levels around kind of a median that you want to keep things at. So that was a bit of a whistle-stop tour. So we can recap that a little bit on Thursday. Have a look through that, you know, in the time between now and next lecture on Thursday morning, and see what doesn't make sense, right? Refer back to the chat, you know, kind of the principles are laid down there. Use the image in front of you, and if you find any good videos or anything like that, then use those too. Pardon me. Anybody need any more time for notes? Cool. All right. Again, my apologies for taking so long in that meeting today. I wouldn't have done so unless it was really important to do so, and it was. So we'll catch up on Thursday. So we'll recap the lac-operon, and we should in the first lecture be able to cover the trip-operon, which is pretty funky in its own right as well. So if you can, have a look ahead and see what you can, you know, absorb from that too. It will help you a great deal in the lecture. All right, everybody, I'll see you in half an hour. Enjoy your lunch and stretch your legs, that kind of stuff. We will be having lecture next Tuesday. Yes. Yeah, so we'll be having, it might be catch-up time. Hopefully there'll be time for asking questions and recapping or going over anything you didn't understand. We'll try and make it all work, okay? Cool. All right, everybody, I shall catch you in half hour.